Morning, everybody. Welcome to Grand Rounds. I have one announcement to make, which is the fact that the third annual Columbia Psychosomatics Conference, which is largely organized by Ala Landa and Brian Fallon, and has been very successful at addressing an area that's really consequential in terms of healthcare, but doesn't get the attention, and there's not the level of sort of scientific information about. But they've kind of shined a spotlight on this through this conference, which I think has been very, very valuable. And it's this third year, presumably will continue, but it's also brought together kind of people with interest from around the area and the country, and thereby hopefully generating critical networks of people to collaborate and dealing with it. So psychosomatic is a broad term. I'm not going to attempt to define it, but apart from more exotic form fruits of classic psychiatric disorders, a lot of it, I think, derives from the 40% of primary care, whether it's pediatric or adult, that those practitioners will say is largely psychiatric. So it's in many ways sort of a less well-characterized area of human disease or dis-ease. And I think Ala and Brian have done a great service by really being leaders in this process. Anyway, so that's going to be October 28th and 29th, and I'm sure there's availability if anybody wants to attend. Also, as you know, we have an Assistant Secretary of Mental Health who was appointed by the Trump administration as a result of the 21st Century Cures Act. It's Dr. Eleanor McCants-Katz, who's an addiction psychiatrist, outstanding individual known to many of us. She's going to be speaking tomorrow at a small conference at Friday, the Manhattan, sponsored by the Manhattan Institute at the Harvard Club. We have a small contingent of people that are going to be going there to hear and to meet with her. But if any individuals beyond that want to go, let my office know, and we'll see if we can arrange it. It's not an open meeting, but I think they do have availability, and obviously they'd like to get the best group of people in attendance as possible. So today we have one of our own faculty to give grand rounds, Martin Picard, and Martin, I think, exemplifies a very innovative, forward-thinking, and uniquely kind of Columbia PI type of faculty member and area of research. He was recruited really as an initiative of the division that Richard Sloan directs, and Richard and his colleagues, who have a broad sort of scope of interest in areas related to behavioral medicine, identified Martin as somebody who was doing work that many people would think of as sort of remote from psychiatry, but clearly was relevant, not just to mental illness, but to basically human biology, aging, functionality. And so it was a little bit of an unusual type of recruitment, but one that made perfect sense when you looked at his CV, heard him talk, read his papers, and thought about it. And through a variety of means, including the support of one of our iconic faculty members and alumni. Should I embarrass him by saying the name, or no? Should we out him? Ralph Wharton. We were able to put together a recruitment package that kept Martin from returning to his native home, Canada, and come to Columbia for at least a period of time, and hopefully indefinitely. So Martin hails from Canada. He was educated at McGill. He received a degree in neuroimmunology at the undergraduate level, and then doctoral level in mitochondrial biology of aging. and then did several postdoctoral stints at McGill and then the University of Pennsylvania as well, and came to us from Penn, where he's now ensconced in the Cole building in a brand new laboratory. And I won't go into the sort of scope of his work other than to say that by studying the physiology of an organelle that's essential to life, to functioning, and its relationship to our functional viability and relationship to the aging process and to disease, it brings a type of expertise in an area of research that is very innovative, very, very potentially contributory and productive, and it's not something that most Department of Psychiatry are sort of featuring or have represented. So really grateful for the Psychosomatic Medicine, Behavioral Medicine Division for having pulled this off, and for the work that Martin's been able to do in the 2 years that you've been here. 2 years? And we know you're not going to forget your French, but your English and New York accent will only get better and better. So please join me in welcoming Dr. Martin Picard.
Thank you, Jeff, for a wonderful introduction, and it's really an honor to be giving grand rounds today, and thank you for the people who've made this possible. It's really an opportunity to present some of the work that we do, and I think Jeff tried to almost justify why we, mitochondria was brought into psychiatry, and I'd like to say that our approach to mitochondria is a little unconventional for the field of mitochondrial biology, and we see mitochondria not as this being powerhouse, which is the typical catchphrase you see in the textbook, but more they're actually living little organisms inside the cell, and they do have somewhat of a social life. They interact with one another, and they divide. They have a life cycle. They respond to stimuli. They produce signals. So I'll show you some data to substantiate this. But really, the idea and our approach to studying mitochondria is one that's inspired by psychiatry and psychotherapy, perhaps to some extent, where we query mitochondria. We want to understand them as holistically as possible, and we have ways in the lab and in the clinic to ask questions to mitochondria. We perturb them, we stress them in this way, and then we see how they respond. So it's really engaging in somewhat of a dialogue with mitochondria with various tools, and molecular biology all the way up to physiology and clinical medicine, that we approach this and study the role of mitochondria in various diseases and in aging process. So everything that I'm going to say today might strike you a bit as a bit mitocentric, and I'll show you some evidence that try to justify that perspective. So the core of our interest is in understanding how psychological stressors end up making their way all the way inside the genome and then influencing disease processes. And there are two papers that I remember reading as a graduate student and a postdoc. That is, we're asking that question: how does psychological stress actually influence physiology and influence clinical outcomes? And there's quite a bit of robust data from preclinical, clinical, and epidemiological studies suggesting that psychological stress does get inside the body and inside the cell, influence things all the way down to gene expression and then clinical outcomes. How does that happen? So we wanted to address that question and position mitochondria in the stress disease cascades. How is it that stress gets translated into disease, and what's the role of mitochondria in this process? So I just want to introduce a term, an allostatic load. I'd like to ask, how many of you are familiar with the term allostatic load? Okay, so the majority. So allostatic load, for those who haven't heard of that term, was introduced in a wonderful paper in 1993 by Bruce McEwen and Steller. And then there's a paper in the New England Journal of Medicine in 1998 that has been cited over 4,000 times because it provided the first framework to understand the neuroendocrine, inflammatory, and metabolic effects of stress on the organism, and then how those changes or this dysregulation can lead to wear and tear on the organism and eventually into disease. So this is, in a nutshell, the allostatic load model as proposed by McEwen and Steller. And the idea is that chronic stress, just trying to get the pointer to work. Okay, so chronic stress leads to primary mediators, and these primary mediators are neuroendocrine and metabolic stress hormones like cortisol and catecholamines, insulin resistance and increasing blood glucose and other metabolic factors. And then the model said that primary mediators are somewhat translated into primary effects, and this kind of remained as a black box. Something happens inside the cell probably, and then that leads to oxidative stress and inflammation, and then leading to primary outcomes. So these are things you can measure in the cell, like cellular dysfunction, telomere shortening, epigenetic changes, changes in gene expression, cellular senescence. And then these primary outcomes at the cellular and molecular level end up leading to secondary and tertiary outcomes that are, typically clinical indicators, from blood work and from various physiological and clinical measures. So the big outstanding question, what are these primary effects? So what we proposed was that mitochondria are actually these primary effects that respond to primary mediators, and then there's something that develops. The mitochondria try to compensate for this stress state by recalibrating, and then this leads to mitochondrial allostatic load. And this manifests in various ways by changes in mitochondrial shape or mitochondrial fragmentation, decreasing quality control of the mitochondria, so you accumulate dysfunctional organelles inside the cells, increases in reactive oxygen species production, leading to oxidative stress. Mitochondrial DNA can become damaged, and then this can lead to decrease in energy production capacity and susceptibility to cell death. So these are operationalized definitions of mitochondrial allostatic load or mitochondrial defect that then we can start to test. So why position mitochondria in such a central place? So there's one argument is that mitochondria are actually at the origin of complex life. So if we take a human cell and we can subdivide it into two major compartments, one of them is the nucleus where the genome lies, and the genome encodes for genes that provide structure. So the genes encode for proteins, and then the proteins provide the structure of the cells and of the organism. There's another compartment, and we'll focus here on mitochondria in the cytoplasm. And mitochondria take the oxygen that we breathe in and the food substrates that we consume and then transform that into cellular energy. The cellular energy currency mostly is ATP. So really the reason why we breathe, and you could go so far as saying the reason we have lungs and a cardiocirculatory system is to bring the oxygen and the food substrates down to the mitochondria. So there's nothing else in the cell that functions meaningfully with oxygen. So we bring oxygen to the cells so that it can enter the mitochondria and allow energy transformation. So the distinguishing factor between a cadaver and a living person, a thinking and feeling body, is really the flow of energy. The molecules are exactly the same number of cells are there, but without the flow of energy through the body, which happens in large part through the mitochondria, there's no life. Mm-hmm.
Question of clarification. Body has many different cell types. Is the function of the mitochondria in all of those the same?
Yes, that's a great question. And I think the next slide addresses this. So the question is, are the mitochondria the same in every cell type? Because there's a lot of different cell types, a lot of different tissues that do very different things. And the quick answer is mitochondria are very different between different tissues. So mitochondria are not all created equal. Yeah. And this has important implications for understanding the effect, for example, of stress on mitochondria or the effect of mitochondrial dysfunction on disease. A very important point is that these two compartments, so the energy flow through the mitochondria and the structure that's provided or encoded by the genes, interact very closely with one another. And there's close crosstalk, and I'll talk about that later. And really this union, or this bringing together of energy and structure was paramount in development of complex life forms. And there's very good evolutionary evidence. If we look at different forms of life, the genus where the mammals are from have much higher amount of energy. So this is picowatts per unit of genome. And this was calculated how much energy is available per unit of genome in different cells, unicellular organisms and then the eukaryotes. And so there's a tremendous difference between the amount of energy and the forms of life that developed multicellularity and complexity like us. And this is in large part accounted by the number of mitochondria and the advent of mitochondria. So if we think about evolutionary history and how we came to be, About 1.2 to 1.5 billion years ago, there's a very critical event that happened, which was this endosymbiosis of mitochondria with the proto-eukaryotic cell. So there was this big anaerobic cell that was not able to use oxygen to make energy, and then this smaller alpha proteobacterium that was using oxygen to make energy, then somehow the big one engulfed the small one. The small one, alpha proteobacterium, was to become the mitochondria, and instead of digesting it and using it for food, they derived some kind of mutual benefit, and that event in evolution enabled the evolution of complex life forms. So in response to Jeff's question, there are mitochondria in every tissue of the body. Every cell of the body has hundreds to thousands of mitochondria, except one cell type, which are the red blood cells. And if you think about it, the red blood cells, their purpose is to transport oxygen to the mitochondria. So you wouldn't want to have mitochondria in the red blood cells that would consume it on its way there. Mitochondria, the amount of mitochondria per tissue is also very different. So tissues that have high energy demand, like the heart and the brain, have high mitochondrial content, and other tissues like the liver and the white blood cells of the immune system have less mitochondrial content. And what do mitochondria do? So we all know mitochondria is the powerhouse of the cell. So ATP synthesis is one of their main functions, but they also do a lot more, including reactive oxygen species production, which is an important signaling molecule, calcium regulation, which is critical in the synapse and neurons in particular, triggering cell death, hormone biosynthesis, so the glucocorticoid hormones, and actually all of the steroid hormones are synthesized inside the mitochondria. So the very first step in steroidogenesis is cholesterol import by a protein on the mitochondrial surface, and then the first enzymatic reaction is a side chain cleavage enzyme that turns cholesterol into pregnenolone. That happens inside the mitochondria. Then it's shipped into the endoplasmic reticulum, comes back into mitochondria, and then the terminal reaction makes cortisol. So there's a very close connection, yes.
Where on that graph would smooth and striated muscle fall?
So smooth and striated muscle, somewhere in between the liver and the brain. Yeah. So if we think about stress as the adaptation, and this concept of allostasis, that the body tries to maintain balance by stimulating different stressors, either in response to real or imagined threat. Every little aspect of the stress response increases energy demand. If you just want to turn on a gene, you want to make RNA from DNA, every nucleotide requires ATP. If you want to make the protein, then every incorporation of an amino acid requires ATP. And then if you want to maintain membrane potential after depolarization, the sodium-potassium ATPases, and so on, muscle contraction. So every little part, every little response to an environmental stimulus requires energy. So if mitochondria are so central to energy production, then maybe they might be implicated in the regulation and of the stress response. And there's this interesting quote from Herbert Weiner in his book in 1992, where he says that stress exposures engenders coordinated and integrated physiological response patterns that are across organ systems that have evolved to prevent injury and promote survival. So we're asking a question as to whether mitochondria are involved in regulating and shaping that coordinated, integrated physiological response. So there's a very important part of mitochondria, which is the mitochondrial DNA. So it's these little circles here in this diagram. This is a magnified version of it. There's 37 genes on the mitochondrial genome, as compared to about 25,000 in the nuclear genome. It's 16,569 nucleotides, so it's small enough that you can actually count all of the nucleotides and name the mutations just by their number. And all of there are two blue genes here, which are the ribosomal RNA to make the polypeptides. There are 22 of these small genes that are transfer RNA, and then the red genes are the messenger RNA, or the protein coding genes. So everything in the mitochondrial genome is there, and all of those genes, their function is solely to make energy. So there's a number of mitochondrial diseases, so the point here is not to go through this list, but every bubble is basically a clinical syndrome or a medical diagnosis that has been associated with a mutation on the mitochondrial genome. The first mutation was identified in 1988 by Doug Wallace, who in 1981 discovered that we get the mitochondrial DNA from our mothers. So we all have our mom's mitochondrial DNA. The father never passes on, which gives more of the power to the woman. And what are those symptoms? So mitochondrial diseases, what kind of symptoms do patients present with? So exercise intolerance is probably the most frequent one, cardiomyopathy, deafness, blindness, diabetes, neurodegeneration, depression, and premature aging. And premature aging, this is just to give you an idea of if the mitochondria go bad, the mitochondrial genome has mutations, this is what can happen to the organism. So these two mice had exactly the same nuclear genome. They're actually littermates. This one is heterozygous, this one is homozygous for the mitochondrial DNA polymerase, for a mutation in the mitochondrial DNA polymerase, that as it replicates the mitochondrial genome and goes around, doesn't have a normal function of the proofreading part of the enzyme. So it introduces errors and mistakes along the replication of mitochondrial DNA. And because mitochondrial DNA are always replicating and degrading, then more mutations accumulate. So these mice have about 10-fold more mutations than their sibling. And as a result, at the same age, they look about twice as old. Their lifespan is half of normal. They get this kyphosis, lipodystrophy, the muscles atrophy, the brain atrophies. And so they get all of these features that are very interestingly similar to human aging. Aged mice don't necessarily look like this, but there's this constellation of symptoms that mimics progeria. Mm-hmm.
A few months ago, there was this publicized case of this kid in England. That was a mitochondrial disease?
Yeah, so the case in England was a case of mitochondrial disease. That was due to a mutation in a nuclear gene that encodes a mitochondrial protein that is important for making nucleotides for mitochondrial DNA replication. So this kid basically had depletion of the mitochondrial DNA. The cells were failing to make new mitochondrial DNA as the old ones were getting degraded. And as a result, this kid had almost complete loss of skeletal muscles, and the brain was severely affected and didn't survive.
I want to get you off topic, but did they make the right decision by denying?
I don't know. Yes. So my colleague Michio Hirano in neurology was closely involved with this case, and there's so many things that happened here, and the clinical picture was complex, and so I wouldn't comment on this. So the only clinical slide I'll present is this, just to give you a sense, and for those who are involved clinically, the kind of presentations that might exist in patients with mitochondrial disease. One syndrome is called chronic progressive external ophthalmoplegia. So this patient, you can see, has droopy eyelids. So here he's trying to open his eyes as much as possible, and here he's trying to look down, look up, look left and right. So there's weakness of the eyes, and the eyes don't move together. So that's this syndrome, and this patient on MRI has cerebellar atrophy. And here's another example of a disease called MELAS, stands for mitochondrial lactic acidosis, stroke-like episodes. So that's quite a mouthful. That was described originally here by Daryl DeVivo at Columbia. And what you see here are these hyper-intense regions on this fluid attenuation inverse recovery sequence by MRI. And these are typical of what you would see after a stroke. After there's an ischemic stroke to the brain, you would see these hyper-intense regions. And you can see the A, B, and C, or this same patient at one time point, and then a few months later, this patient had another episode of symptoms, and then the scan actually looks completely different. And the very interesting thing here is that the lesions, the sites of lesions, don't follow the typical microvascular beds. So this is not ischemic, it's from the mitochondria. So because of the interest of the crowd, I have a slide here, which is a very selective summary of evidence of mitochondrial dysfunction in psychopathology. And so I'm not going to go into this in great detail. And the focus of today's talk is to convey an understanding of the mechanisms by which mitochondria can create these things. But first, let's get an idea of the kind of data that support a role between mitochondrial dysfunction and psychopathology. So there's prevalence data in adults with mitochondrial disease of major depressive disorder, bipolar disorder, panic disorder, and these numbers are quite a bit higher than the population average. Notice that the sample size in this study is quite small, so you can wonder what this means, and also knowing that mitochondrial disease is a medically incurable disease. Being diagnosed with something like this just by itself might make you feel not too optimistic about the future. There's also prevalence data in children with mitochondrial disease, also a small sample size of depressive behavior. There's genetic data in children with autism spectrum disorder that have a high level of pathogenic mitochondrial DNA mutations. So if you take the blood of kids diagnosed with autism, and then you take the unaffected sibling and the mother, so you have the trio, you do deep sequencing, you do whole exome sequencing, you'll find a variety of mutations on the mitochondrial DNA. And then you can compare that to see if the child with autism has more than its sibling. And in this case, yes, kids with autism have more mutations. Those mutations are predicted bioinformatically to be more pathogenic, probably in most cases because they change an amino acid in the given protein. And also those mutations, interestingly, a large fraction of them were not present in the mother, suggesting that they arose during development as opposed to having been transmitted directly. There's also experimental evidence in mouse models where there are mitochondrial DNA defects that can be induced. In this case, it's mitochondrial DNA deletions. So a chunk of the mitochondrial DNA can go missing. Those are frequent in aging tissues in the brain and the muscles. And these mice with these mitochondrial DNA defects showed depressive episodes as evidenced by vegetative and psychomotor disturbances. Very interesting paper in Molecular Psychiatry from last year. And then there's experimental evidence in rats and beautiful work by Carmen Sandy in Lausanne showing that anxiety levels and dominance social behavior are correlated with mitochondrial function in the nucleus accumbens. So the rats that are most anxious actually have different mitochondrial function in the nucleus accumbens than the less anxious rats. And then they went on to do microinjection studies of mitochondrial inhibitors or some molecules that can potentiate mitochondrial function like vitamin B3 and mitoQ, and showed that they could actually influence behavior and social dominance behavior just by injecting and influencing mitochondria, specifically in the nucleus accumbens. And they have this more recent paper suggesting that this, in part, is a result of dopamine D1 receptor activation in the nucleus accumbens. And they've mapped a beautiful VTA to nucleus accumbens pathway and looked at the effect of diazepam. So these are not things that we study, but just to give you a sense that there's emerging evidence. This is very selective, it's not complete, but the mitochondria are likely to contribute to some of the pathophysiological state that we see. So very often, when you want to understand how does something work, and how does something contribute to something else, it's very useful to know what something looks like. So form influences and forms function. So maybe to the younger people in the audience, in this day and age, what do you want to do when you want to know what something looks like? You're sitting at your desk and you're like, I wonder what that looks like. You Google it, exactly, yes. So if you Google mitochondria, this is the picture that emerges. And there's the theme here is that there's these kind of structures. They have this outer membrane, this inner membrane. These are the outer and the inner mitochondrial membranes. And then there's this swimming pool-like structure in the middle that's called the mitochondrial matrix. That's where the mitochondrial DNA is. And you can look at these structures on electron microscopy. So that's what it looks like if you look on Google. Now, if you go into the lab, you have living cells. You put a dye on them that will go selectively into mitochondria to light up the mitochondria. One of those dyes is called mitotracker. And then you look down the eyepieces. This is what it looks like. So here we have a cell. This is the outline of the cell here. This big circular structure in the middle of the cell nucleus. This is where the genes are housed. And all of these squiggly spaghettis in the cytoplasm are the mitochondria. And you can see if you bring your attention here to this quadrant, tremendous morphological remodeling within seconds to minutes. So you'll notice the video is on a loop. The loop is 3 minutes long. So this is accelerated 7 times. But they move fast enough that if you look very closely, you can actually see them move under the microscope. And there's some events that happen. If you look at this mitochondrial tubule here, originally it's one tubule, and then right here, there's a division. So that's called mitochondrial fission, right here. So mitochondria have the ability to fuse with one another and to become longer and bigger, or to fragment to undergo mitochondrial fission. And these shape changes happen in response to stressors and during cell division and so on. And we know now that if we prevent mitochondria from interacting with one another, we disrupt the function of the mitochondria themselves, but also of the cells that they inhabit. So this is the kind of picture, right, that conveys how alive mitochondria are. And if you're going to understand mitochondria, they're not a single biomarker. It's not like a hormone you look at or a cytokine that's higher or lower. There's a complex dynamics. So capturing that dynamics is really what we need to strive towards to understand better the role of mitochondria and various disease processes. So this is a little schematic that I'll use throughout the talk. Here's a cell membrane, the nucleus here with the genes, and then the squiggly mitochondria in the cytoplasm. So really, our main question is, how is it that stressors from the outside, so environmental perturbations, you could say, make their way all the way inside the nucleus? And we break this question down into three main components, so we can ask more precise questions about it. The first is the ability of mitochondria to sense, so their response to these various stressors. The second one is the ability of mitochondria to talk to each other and potentially integrate information. And so that's probably the most speculative part of the model and of the talk. I'll show you some data that substantiate this. And then if a system can sense perturbations and integrate that information, these two things only make sense if then the system can produce some output or some signal that influences the rest of the cell in this case. So the third part is the signaling, and then the ability of mitochondria to generate signals that influence cellular and organismal behavior. So let's focus on the sensing part. So we saw from that movie previously that there's this shape changes in the mitochondria, and that they can dynamically undergo shape changes. How do you look at this? We saw the video. This is two-dimensional. It's in a cell, living on a plastic dish. What if you want to look at mitochondrial shape in a human being? How do you do this? So we've been developing methods for doing this, and here you're seeing Amy Vincent, who is a student and now a postdoc in Newcastle University, and we've been doing three-dimensional electron microscopy. So this is a sequence of EM images, electron micrographs, and then you're seeing at the subcellular resolution, so we're looking highly zoomed in. And then what Amy did was to reconstruct in 3 dimensions the mitochondrial network. So here, each little blob, so each blob of a different color is a single mitochondrion. And we're seeing the collection or the network of the mitochondria in in a patient's leg, actually, from a biopsy sample. And what you can see from this reconstruction is it matches, the typical EM picture that you see with the mitochondrial dyads along the Z lines. So this is in skeletal muscle. We can do the same things in neurons, both in the molecular layer or in the granular layers of the hippocampus, for example, which we're in the process of doing. And really what that allows you to see and to appreciate is the complexity of the shape of the mitochondria. And this one here, you can see there's a big part here, a big part here. There's little connections that bridge these two layers of mitochondria. So this is a single mitochondrion, this complex multi-branched structure. So this is not the textbook bean-shaped picture of the mitochondria, and really trying to understand how these shapes come to be and what their meaning is a new frontiers for mitochondrial biology.
Using the structure, the shape is dynamic, and the morphology varies, but we're not sure in relation to that.
Exactly, yes. So the structure and the shape varies and respond to various signals. We've studied the effect of metabolic oversupply, for example. Very high glucose, hyperglycemia. It's known to cause diabetic neuropathy. It's known to cause brain atrophy over long periods of time. It's been associated with depression. How does this happen? So we've done some work looking at mitochondrial morphology in response to metabolic stress, high glucose, and we find fragmentation of mitochondria within 6 hours in mouse tissues. So I'm not showing this data. Here I'm showing another example. Instead of the stressors coming from the outside, like high glucose, here we're looking at the effect of a mitochondrial DNA mutation on the morphology of the mitochondria. So we're asking if the mitochondrial DNA has a mutation which impacts the ability of mitochondria to make energy, does that change their shape? So here we took biopsies from about 10 patients and 10 controls, and then we did this 3-dimensional morphology reconstruction and quantitative analysis. So this is the first time we can quantify, put numbers on pretty shapes, pretty much. And once you put numbers on those shapes, then you can ask statistical questions. So this is an example of all of the parameters that can be extracted from these models. And you can see that some parameters cluster together. This is just a correlation matrix. Red means highly correlated, blue means anti-correlated. You can see that there are volume metrics that cluster together, shape metrics that cluster together, and then some other ones. You can pull this information into data reduction, or using data reduction approaches like PCA or partial least square discriminant analysis or PLSDA. And here the question is, can we, using the variance in all of these variables, use a two-model or a three-model, a two-component or a three-component model to explain the difference between the groups? Can we separate our groups just based on the shape of the mitochondria? So here you see in this two-component model that you get fairly good separation. In the 3D model, that's slightly better. And then you can ask, well, what's contributing to this? And from this partial least square discriminant analysis model here, you can extract the importance, so the variable importance and projection of this VIP score. How useful is each of these variables in producing that separation? And you find that the proportion of simple mitochondria, so in the 3D models, there were some very complex shaped, and then there were some very simple little beans. So having a lot of little beans in your muscle is highly diagnostic of mitochondrial disease in this case. And interestingly, the second variable here that came out was nanotunnels. So that was a very interesting thing that we observed. So this is an example of what mitochondrial nanotunnels are. So up until recently, these structures had not been described in human tissues. So here there's one central mitochondrion. Here's another mitochondrion, another one. And what you'll notice is that there are these structures that are connecting the mitochondria. The cristae are here, right? The mitochondrial DNA would be in here. And these are these tubular structures. So the first paper to describe this was in 2007 in African green monkey cells in tissue culture. When they blocked the mitochondrial movement, they saw thin protrusions coming out of mitochondria. Then nothing was published until 2013. And then there was a nice PNAS paper in rat heart that showed these connections again. And this is the first finding of nanotunnels in human tissue. And you can ask the question, is there anything exchanged between these? Could the nanotunnels serve the purpose of exchanging information and communication between the mitochondria? So you can do something in the context of, by molecular manipulation of the system, you can photoactivate. So you turn on a fluorophore in a specific mitochondrion, and then you see if that fluorophore can be passed on to another one. So in this movie here, this mitochondrion is photoactivated, and then we're looking as to whether this fluorophore can be transmitted. And then you see there's this protrusion coming out, and then eventually this mitochondrion acquires a fluorophore, and you'll see this one then acquires it. So that provides evidence, and this is very preliminary data, that there's molecular exchanges between the mitochondria. So a little bit, coming back to this idea that mitochondria are not singular bean-shaped organelles that don't talk to each other. They're actually little social entities that exchange information and mix content and so on. So this is one way by which it can do this. And this brings us to the second point of integration. So we'll look at another electron micrograph. This here, what we'll see is a movie. So we're going to go through a thick layer of mitochondria. And there's a few things to notice here. So there's a central mitochondrion here, and then there's another one here that's touching it, another one here, another one, and another one here.
Are those separate, or are those connected to the tunnels?
So those don't have nanotunnels. Otherwise, we would see them between. These are physically touching each other, but they're separate organelles. And at this level of resolution, we can see the membranes. And so these would be like two cells touching each other. So there's a few things that can be noticed here as to the structure of the cristae. This central mitochondrion, for example, there's part of it, the left part, that has nicely organized, more or less parallel cristae. And in the same mitochondrion, this part here has disorganized reticular cristae. The only difference between the two parts is that this part here isn't facing another mitochondrion. This other segment is facing other mitochondria. And a second thing to notice is at these junctions between the mitochondria, if you count the number of cristae that arise from the surface, there's actually a lot more in these areas than here. So there seems to be something about having two mitochondria energized, touching each other, that favors the formation of cristae and their organization. And the third thing, and potentially the most interesting observation, is that if you pay attention to here at this contact, there's actually a coordination of cristae across the mitochondria. And you see them moving with one another. So there's some information being transferred between the mitochondria that makes the cristae of one organelle being aligned with the cristae of the other organelle. So that's the first physical evidence of information exchange between the mitochondria. If we zoom in a bit further, this is a snapshot of with the cristae outlined of the movie we just looked at. Then if we zoom in further on this kind of interface, you can see here this kind of structure. This is the outer and the inner membrane of the blue mitochondrion, the outer and the inner membrane of the red mitochondrion, and this here is the intermitochondrial junction, or the mitochondrial synapse. And you see this cristae is perfectly aligned with this cristae. And if you really trust electron microscopy, you can even imagine that there's a little gap here, a little hole, by which electrochemical information can be exchanged and electrically coupling the mitochondria with one another. And these structures are very similar to gap junctions. And we know now that they're electrical synapses or gap junctions between neurons and between astrocytes and neurons. So we seem to be observing a similar kind of mechanism that allows mitochondria to talk to each other as we see between neurons. So the third component is the signaling part. So that's very well. If mitochondria can change shape and change their function in response to signals, potentially integrate that information, then can they transmit meaningfully that information to the rest of the cell? So there are two arguments to suspect that this is the case. The first one is a topological argument. Where in the cell are mitochondria? This is a snapshot of the movie we looked at earlier. You see that the mitochondria are positioned perinuclearly. That means around the nucleus. And if you zoom in onto this interface, you see here the nucleus in blue. This is the outside of the cell through which the stress hormones, for example, will be coming in. In green is the cytoplasm, and these squiggly red structures are the mitochondria. So if a signal is to come and reach a nucleus, physically it needs to come through the mitochondria. If we zoom in further on this mitonuclear interface, here's what it looks like by EM. So the nucleus, the cytoplasm, and here you see strings of mitochondria attached or connected by these intermitochondrial junctions. And if we look at this junction even more closely, we see this inside the nucleus. So here we're at the level where we see the chromatin. So we're seeing the DNA wrapped into the histones, this dark structure here. And then in yellow is the nuclear envelope. So that's the envelope that packages the genome, that keeps it together inside the nucleus. And these two structures here are the nuclear pores. This is where the mRNA that's made in the nucleus can come out into the cytoplasm. These are the ribosomes, these dark grains. So this is where protein synthesis happens. And then the signals, the transcription factors that are activated by different signaling molecules need to make their way inside the nuclear pores to then activate and deactivate genes. And what we can notice is that mitochondria are very close to these nuclear pores. And that distance, the physical distance of a few hundred nanometers, is definitely diffusion distances for chemical information and signals to make their way inside the nucleus and influence gene expression. So what kind of signals? What are those signals? Well, there's a few epigenetic modifications on the chromatin and on the DNA itself that are fairly well known, and I won't go into this in detail, but we also know the enzymes that are responsible for putting these marks and removing them. And interestingly, all of the substrates and the cofactors that are involved in these enzymatic reactions and chromatin modifications and the epigenome, they're all molecules that are either produced directly or that are secondary metabolites from mitochondrial metabolism. So in a way, mitochondria speak the language of the epigenome. And if you think about, in evolutionary history, how complex multicellular life forms came to be, that was very much tied to the occurrence of mitochondria and energy. So one way to think about this is if you want to regulate which genes is on, which gene is off, you better know how much energy there's around, and you better know what the state of the mitochondria is. So to test that idea, And that's very theoretical. What proportion of the genes in the human genome would mitochondria regulate? So to test that idea, we took cells from a patient that had a mixture of mutant and normal mitochondria. So the green here are the normal mitochondria. The red ones have a mutation in a specific position on the mitochondrial genome. It's a position 3243, so 3243. And it's a G instead of an A. And this causes MELAS. The picture I showed earlier with those areas of hyperintense intensities on MRI. So what you can do with cells from this patient is enucleate them, so you remove the variants from the genetic, from the nuclear genetic material. Then you take a cell line from which you remove the mitochondrial DNA, and then you have cells that are called row 0. They don't have mitochondrial DNA. They're not very happy, but because they're cancer cells, they can survive that. And then you fuse them together. So this creates a cytoplasmic hybrid cell, or cybrid. And then the purpose of this model really is that then you can identify different clones. So you go into your dish, you pick different cells, and then you seed them separately, and then you let them grow. And you can generate clones that have 0% of the mutation. So all of these have the same nuclear genetic material, but these cells have none of the mutation. These cells here at the bottom have 100% of their mitochondria with the mutation, and then you have the spectrum in between. So this is the ideal experimental scenario where you can isolate a single independent variable, the degree of mitochondrial dysfunction when everything else is equal, and then ask what is the effect of the degree of mitochondrial dysfunction on your dependent variable. You pick your favorite here. Here we wanted to look at the transcriptome, so the expression of all of the genes in the human genome. So the first thing we looked at, being interested in the shape of the mitochondria, by confocal microscopy and by EM, we looked at the shape of the mitochondria. You can see that cells with normal mitochondria, they have, some of them are a bit elongated, some of them are bean-shaped. When the mitochondria have high levels of dysfunction, 90% of the mitochondrial DNA here have the mutation, you find that they're much more elongated. And then the row 0 cells, as a control, they have very fragmented mitochondria. So then if you extract RNA, run RNA sequencing on them, so you basically try to read the totality of the product of genes in these cells, then you get this heat map that is a bit of a headache. And then you wonder, what do I do with this? Here, each column is a different gene. There's 17,000 here. And each row is a different cell line, so a different degree of mitochondrial dysfunction. So what you can see here, for example, in the cells with normal mitochondria, red means higher, blue means lower. There's a little group of genes that are expressed fairly highly, and then you see the normal variability across the rest of the genome. Then if you introduce 20% of mutation, you find that this cluster here disappears, but you get a new cluster of genes that are turned on. And then if you introduce 30%, you find this new cluster, 50% here another cluster, 60% different, 90%. Then when all the mitochondria are dysfunctional, the cell nucleus responds to that and then turns on these other genes, and these are the cells here that don't have mitochondrial DNA or are very abnormal. So if we look into specific genes, and here we were interested in the epigenetic regulators, for example, the DNA methyl transferases. These are the enzymes that put methyl groups on the DNA and can silence or activate certain genes depending where the marks are put. So here we're looking at the DNA methylation, DNA methyl transferase 3A over 3B, and you can see that over this increasing spectrum of mitochondrial dysfunction, initially you see that these markers increase but then start to decrease, and then here there's no change from 0, 20, and 30%, and then there's this upshoot. There are other gene markers, MECP2, which has been implicated in Rett syndrome, for example. Here we find that increasing the degree of mitochondrial dysfunction actually increases the expression of MECP2 and other methyl DNA binding proteins. So if we put all of this heat map and we try to understand the difference between the groups, we can use, again, a data reduction approach, here principal component analysis. So we try to see in the 3D space, we try to explain the variance of the whole gene expression profile with these 3 components. The first component explains 25% of the variance, second about 20%, and the third about 18%. And here we position the different experimental groups on this. So here are the cells with normal mitochondria. If you introduce low levels, 20 or 30% of dysfunctional mitochondria, you see that there's this leftward shift, 0, 20, and 30%. And then you would expect, and actually I should say, these cells actually get smaller and smaller. So that gene expression signature and this leftward shift reflects some adaptation or some allostatic process in the cell. These cells get smaller in shape, in size, sorry. And then you would expect from 0, 20, 30, 50 should be here, 60 should be here. But actually we find 50% mutant mitochondria here, 60, and then 90%. These are the cells with all of the mitochondria are dysfunctional, and these are the cells that don't have mitochondrial DNA. So these are in different worlds completely. But what emerged from this is that changing a single nucleotide in the mitochondrial DNA, 3243, changing it into a G instead of an A, can transform the whole transcriptome. And the cells with exactly the same nuclear material growing into exactly the same environment acquire very different characteristics just because we changed the mitochondrial DNA. So that was a proof of concept study that demonstrated how much influence mitochondrial signaling could have on the transcriptome. And I should point out as a little summary that here over 65% of the human genome was influenced and resulted in these signatures. So that's more than half of the human genome potentially under regulation of mitochondria. So I just showed you this evidence on signaling, and I'll draw a parallel with the brain. And to summarize the sensing, the integration, and the signaling, and there's this quote also from Herbert Weiner's book, Perturbing the Organism, where he says the organism is integrated into a larger system of information exchange. And he points out that the brain and the rest of the organism are not qualitatively different in their ability to compute information, but show only qualitative differences in their purposiveness. So I think what Herbert Weiner is saying here is that information is processed and integrated not only in the brain, which is a fantastic place to do this, but also in other parts of the body that can respond to various signals. So if we think about how neural networks within the brain sense information, and then that information can be integrated and processed through the action of neural networks, and then there's synaptic plasticity and memory formation that kind of sustain that and allow those processes to persist in time. And then the purpose of sensing and integrating that information is then to generate intelligent neuroendocrine outputs and regulating and coordinating the organism's function. So now that we're discovering that mitochondria or have the ability to communicate with each other, we can ask the question, does this give them some similar potential as neurons? And as far as we understand, neurons don't hold a memory. You don't have the memory of your grandmother in a single cell. We hold memories in neural ensembles and through the collective action of pathways. Same thing for processing of information. So if mitochondria can exchange information, then can they engage in some form of processing? So we can think about the mitochondrial network, their ability, like I've showed you, to integrate some information, undergo shape and function changes, then maybe do some kind of signal processing and information, and then finally producing outputs that regulate gene expression and potentially other organismal functions. So now we'll switch gear. So this is kind of setting the layout and the ground information, and I'll show you two pieces of data. One is a human study where we looked at the effect of psychological stress on the mitochondria, and the other asked, well, once the mitochondria are dysfunctional, can they influence how the organism responds to stressor? So if we look at psychological stress, and then we look inside the mitochondria, we can ask, in humans, does psychological stress change the mitochondria? Does that cause mitochondrial dysfunction? And so we performed a systematic review to evaluate evidence in the literature for this. So at the end of this process, we had 23 studies. And I'm not going to go into this in detail, but this is the overall picture. 23 studies, none of them in humans. Here we're looking for experimental studies that manipulated psychological stress and looked at a defined aspect of mitochondrial function. All of them are in male animals, which is not quite acceptable if we are to understand the effect of stress on the mitochondria. So there's definitely progress to be made in this area. So we wanted to look at this in humans, and there's not really a good way, a good platform to measure mitochondria in health in humans. So we isolated leukocytes, and then from the mitochondria, if we look at the inner mitochondrial membrane, this is where the action happens in the mitochondria. And not to bore you with details, the food substrates that you eat are metabolized by enzymes here in the TCA cycle. Then the electrons are transported into these proteins, complexes 1, 2, 3, 4, 5. And then at complex 4, this is where the oxygen that we breathe actually is consumed. So we breathe to power complex 4, and then that allows the pumping of charges across the membrane. The mitochondria are charged like little batteries, and then they use that to power ATP synthesis through the ATP synthase. So here we're focusing on 4 specific parts of the mitochondria, complex 2 and complex 4, which are energy-producing aspect of mitochondria, an enzyme here called citrate synthase that is a marker of mitochondrial content, and then mitochondrial DNA, which reflects also how many mitochondria there are. Yes, so leukocytes are used because they're convenient and because we can then scale this approach into a larger number of people. Ideally, we would look at different tissues. Yes, exactly. Yeah. So then we take those 4 measures and we integrate them mathematically into a very simple equation. We take the 2 energy-producing components, complex 2, complex 4, we add them up as a numerator. And then at the denominator, we take citrate synthase and mitochondrial DNA copy number, which are markers of the capacity of the mitochondria, the content of the mitochondria. So here you have capacity divided by content. So if you have a few mitochondria, so a small denominator but very high capacity at the numerator, then you have healthy mitochondria. That's the idea. And we compared this between controls and caregivers in a study with Alyssa Epple. And here we're looking at how well these different markers, these 4 markers, and is the combination of these 4 markers into this mitochondrial health index useful to separate the effect of stress in these 2 groups. And what we find by looking at this variable importance and projection score, how useful each parameter is that the mitochondrial health index is a superior index. So this is what the distribution of the mitochondrial health index is in this population of here 85 women. So you can see that it's more or less normally distributed. There are values that start at 55. The highest is about 170. So the first thing that struck us here is this mitochondrial health index is hugely variable between individuals. So some people have a health index that's 3 times more than other people. And this opens the door for mitochondria as being potential regulators of inter-individual differences in stress resilience and adaptive capacity. And the second thing to notice here is the difference between the 2 groups. So the caregivers which score higher on anxiety, on depression, and lower on mood metrics have significantly lower mitochondrial health than the controls with an effect size that's moderate and significant. A very interesting thing that we wanted to look at here, and that was possible because of the design of the study by Alyssa Epple, where they measured mood and psychological states at every day for a whole week. And then they took blood here on day 4, where we obviously took the cells to measure mitochondrial function. So here we can ask, is mood on the days before, days 1, 2, and 3, correlated with mitochondrial function, or is mitochondrial function correlated with mood afterwards? And that would give us some indication of the directionality of this effect. And what we find is that if you take the week average, there's a marginal correlation between how people feel over that whole week and the health of their mitochondria. If you take the days before and you ask about positive mood, there's a significant correlation. Here we're looking at the correlation coefficient of the effect size. So this is strongly or significantly correlated with mitochondria, but the days after are not. And you see the effect for negative mood, which are weaker. If we ask those questions at night, it seems like positive mood at night, especially on the days before, are more strongly associated with mitochondrial health. This is breaking down the data on a per-day basis. So here, the higher on this y-axis, the stronger the correlation between the mood and the mitochondria. So you see for the positive morning and nightly, as we get closer to sampling, taking the mitochondria from the participants, the correlation between the mood and the mitochondria climbs. And then it kind of regresses to the means afterward. And if we can look at this in a slightly different way, by tertiles of mood, so people who score low on negative mood have the highest mitochondrial health, and then moderate and high, you see that there's a dose effect of mood on mitochondrial health, and for positive mood, you see the opposite effect. So this really is preliminary evidence suggesting a directional effect of mood and psychological states on the mitochondria. And this opens up a lot of questions that we're interested to understand. So for sake of time and to keep time for discussion, I think I'll skip the next portion, and then there can be more time for interactions. So I'm just going to say that now we're in the process of taking some of the preclinical data that we've collected previously, where we generated mice with transgenic mice with dysfunctional mitochondria, and then we ask, in these mice that have abnormal mitochondria, if we expose them to psychological stress, do they respond differently? And we found that each defect of mitochondria that we introduced generated a unique pattern of stress response. And we looked at HP axis, so cortisol release. Some mice with mitochondrial dysfunction had, twice as much cortisol being produced, but a completely blunted sympathetic nervous system response. And so these unique differences led us to believe that maybe mitochondrial dysfunction is responsible for how each person's physiology behaves in response to psychological stress. So we're taking this study now in humans. So we have some pilot funding for that study called the MISB study, Mitochondrial Stress, Brain Imaging, and Epigenetics study, where really what we want to do is to look at the effect of mitochondrial dysfunction. And we have groups of patients with mitochondrial disease, and then we're going to look at the stress regulation via allostatic load and epigenetic regulation. And we're asking the question using MRI whether changes in brain activity and functional connectivity between specific brain regions are mediating that effect. So is the effect of the mitochondria on the abnormal physiology through the brain because the mitochondria are affecting the brain, or is it because they're affecting other organ systems? We have a fantastic group of people that have been contributing to the development of the MISB study. Michio Hirano in neurology sees patients clinically with these mitochondrial defects. Tor Wager, who was at Columbia in psychology a few years ago, now is at Colorado Boulder, and he does functional neuroimaging. Francis Champagne is a developmental epigeneticist. Todd Ogden, who's here in the audience, is in biostatistics. Richard Sloan, chief of our division, and Bruce McEwen, who's kind of been grandfathering the development of this project. So to summarize, I think if you look in the textbook and what I was taught as a undergrad and grad student is, there's the almighty nucleus that then determines how everything in the cell functions, the mitochondria and different structures and specificity in the specialization, and then mitochondria produce energy that then animate the structure and specificity. And some of our work and others is suggesting that actually the mitochondria can influence a lot of what happens in the nucleus, and that the environmental stressors might actually affect the cell nucleus and the rest of the organism through its effect on mitochondria. So if we pose this question in a slightly different way, as a gene-environment interaction effect, what is this X factor? And we think the mitochondria is the X factor in this gene-environment interaction. So in summary, I've told you about some of the evolutionary process that made complex life possible and how mitochondria were involved in this. Showed you how mitochondria are dynamic little living organisms inside the cells. They're in close proximity to the nucleus and can change gene expression. I showed you some data that psychological stress might predict future mitochondrial health in humans, and we need definitely more work to be done on that front. And this is the mouse work that I'm happy to discuss, and that is published, showing that changing the mitochondrial function in the mice changes the stress response signatures. So we're in Kolb 4, as Jeff said, in the fantastic new lab space that we share with the Salser lab and the Marks lab. If you want to see mitochondria, feel free to come on Kolb 4. This is the group of people who are now taking these studies to the next level, and there are many of them in the audience today, so I won't we'll save time for questions. And these are the collaborators that made a lot of the work that I showed possible. I was a postdoc in Doug Wallace's lab. I did some work there. Caroline in the lab has been collaborating with colleagues on different projects. The development of our mitochondrial phenotyping platform, where we're developing the capacity to assess mitochondrial health in different populations, was developed in collaboration with Jan Burel. There's a group at UCSF led by Alyssa Eppel, where we've done the caregiver study with. Fantastic group in Newcastle, where we've been doing the three-dimensional EM work and some mitochondrial disease work. We work with Yuri Haniowski at Thomas Jefferson on mitochondrial dynamics. And this is our fantastic MISB core investigators. And there are also a lot of people that don't fit on this slide, but that are making this study possible, people working with Richard, and so on. And I'd like to say a special thank you for Richard, who's the chief division, for making this lab possible, and I'm really delighted to be here. Yes, thank you.
Martin, thank you. That was really brilliant. It was really fascinating sort of presentation on mitochondrial biology. I couldn't help thinking that, you know, like in the brain, you know, the glial cells get short shrift relative to neurons, and I guess in terms of cell biology, mitochondria get short shrift in relation to the nucleus and other organelles. But just one sort of very practical question. If you're, apart from just mapping or characterizing mitochondrial biology, if you're relating it to human disease or human sort of health states, if you're using whether it's leukocytes or fibroblasts or anything, and the particular condition you're measuring is from a remote organ, is there, I mean, how do you sort of understand the possibility that it's not really reflecting the consequence that's occurring in the organ of interest that's mediating the disease or the behavior of interest?
Yes, that's a fantastic question, and we were just talking about that with Ralph earlier. This is a question that gets at the ecological validity of the leukocytes for the whole organisms, or some ecophysiological validity. And there's some evidence, partial, but changes that happen in the mitochondria of the leukocytes reflect or are correlated with changes that happen in the brain and in the muscle. So there's a few papers suggesting that even though mitochondria are different in these different organs, they respond in some kind of coordinated fashion to specific stressors and to the specific disease states. One example, quickly, is an Alzheimer's disease, where we find complex 1, the different complexes of the electron transport chain. There seems to be a specific decrease of complex 1 activity, which is also seen in the leukocytes, interestingly enough, even though the disease manifest and the phenotype is mostly about a brain phenotype.
Brian Fallon back here.
Thank you. That was a totally fascinating talk, beautifully presented as well, great images. So I have two questions and a gazillion other questions, but I'll just ask you two. One was, when you talked about evolutionary development of the mitochondria from alpha proteobacteria, it made me wonder whether mitochondria, because they have bacterial progenitors, might be vulnerable to damage as a result of antibiotics. So that was one question. And the second question is, which is partially related to that, is that there was a paper that came out in 2016, I think it was PNAS, talking about chronic fatigue syndrome and mitochondrial dysfunction by looking at metabolomics, which is a step removed from looking at the leukocytes, as a reflection of mitochondrial dysfunction, suggesting that there's something called the Dower response or cellular damage response or cellular protection response. I forget what it was, but maybe you could comment on that?
Yes, definitely. Two great questions. The first one about antibiotics. We actually, by antibiotics or anti-bio, right? Anti-life, and they're targeted specifically at components of the bacteria, mostly that enable bacteria to replicate their genome and to make proteins. So they're mostly DNA replication or protein synthesis inhibitors. The bacterial gene expression and protein synthesis machinery is very similar to the mitochondrial machinery for mitochondrial DNA replication and protein synthesis. And there's actually very good evidence, particularly in kids with some mitochondrial DNA defect and that are predisposed in some way, giving them certain antibiotics can make them deaf. And there's some fairly well documented cases of that. And for some reason, the hair cells in the inner ear would be more susceptible to that. Great question, and we know very little about that. And some people think that most of the side effects of the antibiotics are because of damaging effects on the mitochondria. Regarding the second,
Go ahead. I'm sorry.
remind me the second question again. Yes, chronic fatigue syndrome. Yes, metabolomics. So the metabolome, the collection of all of the metabolites in the blood, is in large part a reflection of mitochondrial metabolism in various tissues. Everything kind of gets dumped and gets taken away from the blood by cells across the organism and mitochondria. When the mitochondria is dysfunctional, that results in a change in the metabolome. So there's other evidence than this metabolomic study that chronic fatigue syndrome implicates mitochondrial dysfunction. So we're just scratching the surface of this, but there's some investigators are fairly convinced mitochondrial dysfunction is at the origin of that, and that's substantiated by some data.
Terry and then Jay.
Thanks very much for an informative talk, and I recognize that you think the cell is there to keep the mitochondria dry in the rain. But really, who's in charge of aging? I didn't get that from your talk. So if you put young mitochondria in an aged cell, or the reverse, which would have a bigger impact? Because you could say, well, the cell is signaling, or the cellular functions are signaling the mitochondria to over-replicate or to change their morphology in aberrant ways.
Yes. I think that's a question we've thought a lot about, and Gabriel in the lab is doing some experiments to look at the role of mitochondria in human cell aging. I think there's evidence to support both sides of the coin. Some of the accelerated mouse model I showed you, if you introduce mutations in mitochondrial DNA, those two papers in Nature and in Science, 2004, 2005, were the first to provide evidence for this causal relationship. If you damage the mitochondria, you cause aging of the organism. And there's more papers since then that have substantiated that. But then there's also, if you think about reprogramming, making iPSCs from very old cells, you can make them young again. And there's evidence that the mitochondria are very different between stem cells and differentiated cells. So I think our paradigm, the way we think about this, might not be complete. So there's some unresolved question there, definitely. I don't have a better answer than that.
Just a simplistic follow-up question. The studies that were done about transfusions of young blood to older animals, how does that reflect what Terry?
Parabiosis.
Yes. Yeah. So parabiosis, and if you connect an old animal to a young animal, you can connect their circulation, so they start to share the same blood. You can make a lot of features of the old animal go back to being young again. So that suggests there's something shared in the circulation, some soluble factor or whatever it is that can be transferred. We're starting to understand more and more about mitokines, and mitochondria can actually release material. There's evidence that mitochondria release mitochondrial DNA, and Caroline, the postdoc in the lab, is working on this, and Kalpita. We're trying to understand how that happens, and there's a lot of soluble factors released by mitochondria, and that mitochondria can trigger expression of in the cell nucleus, like FGF21 is a fairly well-known one that can have broad metabolic effects. So if you damage mitochondria, you induce some genes in the nucleus that make proteins that make their way into the blood and then can transform the function of virtually all tissues. So that's one potential mechanism by which mitochondria influence not only their little confined cell, but the rest of the body.
Jay?
Great talk. So just going back to the evolutionary part, there was the question that Brian asked. It occurred to me that the genome of the mitochondria really does look bacterial. It looks like it's intronless. Is that true?
In what?
Intronless. It doesn't have introns.
Correct. Yes.
And I'm guessing also it's therefore not really susceptible to the same regulatory mechanisms such as DNA methylation or other it really doesn't form chromatin.
Correct. So the mitochondrial DNA is circular. It's a plasmid, just like bacterial DNA. So that's one more ancestral or vestige of their bacterial past. And it doesn't have introns, so that's probably why there are so many mutations in mitochondrial DNA that are that cause disease. Because if you make a change, chances are in the nuclear genome there's 98% non-coding. It's going to fall somewhere where it doesn't really matter. In the mitochondrial DNA, 98% codes, and there's only a very small part where the replication starts in the D loop, the origin of replication, where there's no gene that we know of. So yes, it's very dense, doesn't have histones, and that's also why potentially it's more vulnerable to oxidative stress and to accumulate mutations and deletions, because it's not protected by these surrounding proteins. And the repair mechanisms, the replication, the transcription, and the repair mechanisms are completely different. Different proteins, different genes code for the mitochondrial mechanisms and the nuclear mechanisms.
And I guess the other part was just the symbiosis between the mitochondria and the hosts, because obviously a certain percentage of the makeup of the mitochondria comes from the eukaryotic genome.
Yes, there's...
And how is that regulated?
That's a great question. So this issue of mitonuclear communication is a very topical one right now in the field of mitochondrial biology, understanding how mitochondria talk to the nucleus and how nuclear changes affect the mitochondria. And there's definitely a two-way stream. Mitochondria have about 1,200 to 1,500 proteins. Only 13 are encoded in the mitochondrial DNA. So, it's 1% or 2%. So you definitely need a lot of input from the nucleus for the makeup of the mitochondria, but mitochondria have a lot to say about expression of genes in the nucleus that inform this makeup. So there's definitely a bidirectional communication happening there, and yeah. So there's an important symbiosis. There's this beautiful paper, just to digress just slightly, in human fibroblast PNAS 1979, I think. They took human fibroblasts, enucleated them. So you can do this by centrifugation and cytokinesin B. You remove the nucleus out of the cell. You put them back in the dish. Then you would think, well, the nucleus is the brain of the cell. Huh? The cells are not going to know what to do. They attach, they migrate, they exhibit contact inhibition. So they move, and when they touch each other, they start to go into a different direction. They survive, and they live happily. The only thing they cannot do without the nucleus is to divide. But other than that, cell behavior is completely the same without the nucleus. If you do a similar experiment and you knock out the mitochondria, pharmacologically, the cell dies in a few minutes. So again, maybe I'm biased by this mitocentric perspective. Yes.
Pat, there's a question in the middle of the row. But before you ask, I'm just going to interject one thing. How telomeres have been focused on as being some kind of bioindex or biological sort of measure of aging or sort of viability. Do you see the metrics that you use to gauge mitochondrial health as having a similar potential?
So that's something we're very interested to look at. If you perturb the mitochondria, do you change the telomeres? There's only one paper that I'm aware of that has looked at this in humans. That was from 1997, nothing since then. In patients with mitochondrial disease, their primary defect is this mutation, the 3243 that causes MELAS. Their telomeres were about half of normal. So I think there was a 12 and a 14-year-old boy that have telomere length, the same age as an 85-year-old subject. So there's evidence in that direction. If you perturb the mitochondria, you perturb the telomeres. There's also nice tissue culture experiments on this. But there's also evidence the other way around. So if you make telomeres much shorter by knocking out TERT, the catalytic subunits of telomerase, the telomeres become shorter in mice, and then as a result, that inhibits one of the transcription pathways that is responsible for making more mitochondria, called PGC1 alpha, and then the mitochondrial function diminishes as a result of telomere erosion. And recently at NIEHS, there was a workshop to look at exactly this question, stress, telomeres, and they were interested to consider mitochondria as a potential mediator of that relationship. And obviously, we think that might be part of the story.
Yes.
So all of the data that you were showing earlier linking the mitochondrial response and stress are in endothermic organisms. And so I was wondering if you could speculate on how the situation might look different in exothermic organisms where mitochondria are playing a very different role in energy metabolism.
Yes, fantastic question. There's there's a paper out there that's under review in Science, and it's on bioRxiv, so you can go and see it, showing that mitochondria, the temperature of the mitochondria is actually 56 degrees Celsius. So we know core body temperature is 37. So if the mitochondria are at 50, close to 60 degrees, that is supporting previous, older physiological evidence that mitochondria is actually the reason why we are warm. And the source of heat. And there's something called the uncoupling protein And then in rodents, they have a lot of brown fat. There's a patch of brown fat subscapular, and that's how small animals regulate body temperature, by uncoupling the mitochondria. So they basically uncouple the respiration of the mitochondria with energy production. Then they're no longer making as much energy, but they're making a lot of heat. So mitochondria definitely contributing to body temperature regulation, maybe by being so warm as 60 degrees. There's not a lot of work on exotherm animals, so I don't know. It's a good question.
Alana, what rotation are you on now? Child psychiatry? I was going to say, I was wondering how you got time to get away to come to Grand Rounds, so. David?
So the mitochondria, this is really a very spooky story. I mean collectively they're kind of a Lamarckian homunculus running everything. And what I'm interested in is that they seem to be aware of so many things. And I guess the list of things that mitochondria can sense and be aware of is growing. So is one of the things that they can sense a magnetic field? Because many tiny organisms do.
Yes, and there. So we've thought about this a lot, and thinking about mitochondria, perceiving the environment. What's the limit of that perception? So it's probably not limited to chemical cues, and there are magnetotactic bacteria. So there's evidence that some bacteria respond to magnetic fields. There's evidence that mammalian cells respond to magnetic fields. So we don't know about the mitochondria. We saw this video of the cristae that are aligning. Yes. How does that alignment take place? It's very hard to imagine a molecular mechanism for this to happen across a distance and across 4 membranes. The most likely explanation, if we go for parsimony, is that there's some electromagnetic field generated in the cristae. And if you go back to physics, every time there's current, ions flowing, there's a magnetic field, the right thumb rule. So there are a ton of electric current happening in the cristae. So they're, just by... That's a great question. Some of the preliminary work that we've done, and I know has been done in other labs, show that mitochondria respond to electromagnetic fields only in a very narrow spectrum of strength and pulse. So the MRI is probably outside that range, so people can do MRI safely without having their mitochondria wrecked.
David, don't give the personal injury lawyers any ideas.
Hello. Hi. Awesome talk, and welcome to psychiatry. I love this biology being, highlighted. I also have many, many questions, but I'll leave it at this. So is mitochondria actually an independent cell, a network, a multicellular organism within cells? And if you take it out of a cell, just put it in cell culture by itself, will it survive and replicate? And then, if that's the case, necessarily, I would think that if in cells you have many single-cell bacteria running around, that none of their DNA is the same, actually. That so you have many different kinds of cells within each cell.
Yes. We think mitochondria are a multicellular organism inside the cell. If you take them out, however, they do not replicate and they do not survive. They need the cell. They need this context to exist, which epistemologically is very interesting. There's a lot in this area that we don't know. So yes, there's a lot of experiments that would be very interesting to do, but we haven't done them.
It's good to see that some of our residents' questions can stump the experts, so just to keep you on your toes. So Barry, a lot of this, if not most of it, sort of rolls up to the whole, area of aging, and seems very relevant, even though it's not been exploited as much as it should. You have any comments on it from the context of your perspective?
I don't think I can. This is at a level that I can't do, but it's fascinating.
Use the microphone.
It's opened up a whole panorama of possibilities, thinking, and findings. One's terribly impressed. But to transpose that into the area of aging, I think is premature. Certainly it has a bearing, but what the bearing is, we don't know.
Myron, since you're sitting nearby, do you want to chime in as well? Myron Hofer? Myron? I'm inviting you to comment since you're sitting nearby also.
I'm thinking of lunch.
Feeding the mitochondria.
Touche. Okay, well, wait a minute. We got to let Ralph have the last word.
Well, I wondered, you have a slide about the number of papers on mitochondria, which might impress the audience. You want to show that. But my other question was, you want to say something about mitochondrial transplants?
So mitochondrial transplant. So there's a, if the mitochondria are so good and important, and you have a dysfunctional mitochondria within a cell or within an organism, it might do something good to actually take viable, healthy mitochondria, inject them into the brain or into the circulation or into a cell. And, so that idea of the mitochondrial transplant has been tested and so far has been shown to have some promise. There's been experiments done in the heart after myocardial infarction. There's been some experiments, I think, preclinical models in the brain. And there's even a study from a Chinese group that you take with a grain of salt where they injected mitochondria in the circulation, mitochondria in the blood, and they find that the mitochondria are actually incorporated, become intracellular in various tissues, including the heart and the liver, and enhance mitochondrial capacity in those tissues. I don't know what to think about that, but that reflects those kind of ideas. I think reflect the other thing you were alluding to. This graph. I don't have the slide. I'm sorry. I should have had it as part of the other slides. The number of publications and across the biomedical sciences on mitochondria or on the nucleus or on other organelles, and there's this huge rise of papers around the nucleus, that actually rose all the way up to 2001 when we completed the Human Genome Project, and then we thought we would find the gene for X, Y, and Z, and we didn't. And then the publications about the nucleus are on the way down, and the mitochondria are on the way up. And now there's more papers published about mitochondria in the biomedical sciences than the other organelles. So I think that's what you were referring to. Yeah.
What's one last thing before we close? Maura, in post-mortem analyses, has there been a focus on mitochondria and different types of mental disorders at all?
Well, not that much. We actually already had discussion about this, but we didn't have a formal meeting yet. We discussed the possibility to try to start doing that. I've been studying aging in different ways, looking at, there is a paper under review now, and we look mostly at angiogenesis, vascularization, and neurogenesis, and it seems, but I don't know too much about.
Yeah, but I mean, given the fact that you've demonstrated these morphological changes and the the plasticity of mitochondria, which obviously,
Right.
the fact that it's post-mortem tissue, would limit in being able to
Yeah.
characterize. It would seem to be that would be an interesting focus.
Yeah, we were planning to meet.
Yes, we should talk more.
Yes. We have one more reason to do that. Okay.
Well, listen, Martin, keep up the great work. It was fascinating to get this update. And Richard and Ralph, kudos and thanks for bringing Martin to us.
Thank you.